The topic for today's lecture is nucleic acids. The checklist for this lecture is: in this lecture, we will study the definition of nucleic acids. We will study the introduction of nucleic acids. We will study its structure. We will study the discovery of nucleic acids. We will study the classification, and in the end, we will study the functions. First of all, we will start with the introduction of nucleic acids. Friends, nucleic acids are present in all living organisms, be it plants or animals. Nucleic acids are made of the following elements: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Now we come to the discovery of nucleic acids. How were nucleic acids discovered? Friends, first in 1861, there was a scientist. He isolated nucleic acids. The name of this scientist was Friedrich Miescher. He isolated nucleic acids from the pus cell. Then he named it nuclein. But later it was found that the properties of nuclein are acidic. And since its properties are like an acid, that is why its name was changed to nucleic acid. And this is how nucleic acids were discovered. Now we come to the structure of nucleic acids. Friends, nucleic acids are made up of nucleotides, and nucleotides are the monomers of nucleic acids. Just like I previously taught about the monomers of proteins, lipids, and carbohydrates. What are they? For lipids, there are amino acids. Now we come to nucleotides. Nucleotides, which together form nucleic acids, are themselves made of nucleosides and phosphoric acid. This means phosphoric acid and nucleosides combine to form nucleotides, and nucleotides combine to form nucleic acids. Nucleosides themselves are made up of nitrogen bases and phosphoric acid, that is phosphate sugar. Friends, nitrogen bases, ribose sugar, and phosphoric acid. These three together form a nucleotide. To explain it shortly, a chain of nucleotides forms, meaning nucleotide, 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 all combine to form nucleic acid. This is how the structure of nucleic acids forms. I just explained that nucleotides combine to form nucleic acids. This means nitrogen bases and phosphate sugar combine to form nucleosides. Nucleosides combine to form nucleotides, and nucleotides combine to form nucleic acids. And the nitrogen bases are of 5 types: adenine, guanine, cytosine, thymine, and uracil. Friends, adenine, guanine, and cytosine, these 3 nitrogen bases are present in both DNA and RNA. But thymine is present only in DNA, and uracil is present only in RNA. These 5 types of nitrogen bases are found in nucleic acids. Now we come to the difference between DNA and RNA. Friends, I have already made a separate lecture on this. You can watch that. Its link will be given in the description. The difference between DNA and RNA, their functions, classification, and types. Now we come to the classification of nucleic acids. The first classification is mononucleotides. Mononucleotides are the first classification. Then the second classification is dinucleotides. The third classification is polynucleotides. Let us study these one by one. The first classification, which we call mononucleotides, it is made up of a single nucleotide. Mono means one, and it is always found single in the cell. Its example is ATP. If asked what is the full form of ATP, ATP is adenosine triphosphate group. This means it is made of adenosine and 3-phosphate groups, meaning 3 phosphates and 1 adenosine combined to form ATP. As I already explained, adenosine is a nitrogen base. What is ATP? ATP is an energy-rich molecule. It is a molecule of energy. That is why it is also called the energy currency. This means for any function in our body, energy is needed. That energy currency is ATP. Just like in daily life, we use money to buy things. In the body, energy currency is needed to buy anything, and that is ATP. Now, how is ATP formed? When one ATP is converted into one ADP, then energy is released, and that energy is used in various functions of the body. If we talk about energy, when one ATP is converted into one ADP, the energy released is 7.3 kmol. This is also called 71.81 kJ. Now we come to the second classification of nucleic acids. Dinucleotide. Di means two, and nucleotide means nucleotide. When two nucleotides combine, it forms a dinucleotide. Its example is NAD. The full form of NAD, nicotinamide adenine nucleotide. What is NAD? NAD is a coenzyme. I explained coenzymes in the previous chapter on enzymes. This NAD helps in removing hydrogen atoms from the substrate. Now we come to polynucleotide, the third classification of nucleic acids. Poly means many and nucleotide means nucleotide. When many nucleotides combine, it forms polynucleotides. Examples of these are DNA and RNA. Friends, I have already taught about DNA and RNA. DNA is the hereditary material, and RNA acts as a carrier of information. This means RNA carries information from one place to another in the cell, and DNA is transferred from parents to children. Question of the day. What are the building blocks of nucleic acids? Only a genius can answer in the comments below.